everyone, I am Kia Joy D. Palorma from BSN 1D. In today's video, we are going to explain to you how viruses invade cells. Second is, why are there multiple COVID-19 vaccines existing? And lastly is the difference between the traditional COVID-19 vaccines from mRNA.
Viruses are tiny infectious agents that rely on living cells to multiply. They may use an animal, plants, or bacterial host to survive and reproduce. As such, there is some debate as to whether or not viruses should be considered living organisms. A virus that is outside of a host cell is known as a virion. Viruses are perfect parasites. It has been known for decades that once a virus gets inside a cell, it hijacks the cellular processes to produce virally encoded protein that will replicate the virus genetic material. Viral mechanisms are capable for translocating protein and genetic material from the cell and assembling them into new virus particles. Contemporary research has been revealed specific mechanism viruses use to get inside cells and infect them.
How viruses invade cells. It is becoming clear that viruses have acquired an ability to exploit this mechanism to invade cells and facilitate their own life cycle. In obesity and diabetes, these mechanisms are chronically activated due to the deteriorating metabolic state, and this may provide an increased opportunity for a more profound and sustained viral infection.
I'm Anna Minala, and there are multiple COVID-19 vaccines existing. There are several different types of vaccines that are used to train your body to fight an infection. You may have heard of Pfizer and Moderna vaccines, refers to as mRNA vaccines, and AstraZeneca and Johnson & Johnson's vaccines are adenovirus vaccines, and also the Sinovac, Sinopharm. All types of vaccines work to teach your immune system to respond to an invader, but it differs in the method by which it trains your body to fight this invader. When your immune system meets a viral threat, it begins fighting with a generic response, which is not specific to any particular virus, but over the first 1 to 2 weeks after infections, your adaptive immune cells kick in. These cells are specialized for the particular virus you are fighting. They can learn to recognize the specific structures of the virus called antigens and train to effectively kill them. Some of these cells produce antibodies, which you've probably heard a lot about lately. Antibodies are large proteins created to target and stick to the antigens on the virus and help to kill them.
Good day everyone, I am Stephen Puliga. Today I will tackle to you the different types of vaccines to better understand how the different vaccine works. First, killed weakened virus vaccines. This method is the oldest and the most traditional, involves using the virus itself as a vaccine. Basically, this technique introduces the virus into the body, triggering the body to produce antibodies and memory cells pretty much the way it would do with an infection. Second, the viral vector vaccines. Memory cells and antibodies form to target the antigen portion of the virus. However, the antigen alone doesn't make you sick because it's just one small portion of the virus. This means that you could somehow introduce the antigen into your body without the rest of the virus. You might be able to get your body to make memory cells without getting sick. Third is the protein-based vaccine. Finally, we have more direct approach for introducing antigens into the body. A protein-based vaccine which directly provides your body with SARS-CoV-2 antigens. This straightforward approach avoids obstacle in the delivery of viral vector and nucleic acid vaccines. And because only a non-infectious portion of the virus is added, there is no risk of infection. Fourth is the vaccine regimens. As of its initial approval, the Johnson & Johnson vaccines require only one dose, while the Moderna and Pfizer vaccines follow a two-dose schedule. For Pfizer vaccines, the interval is 21 days between the first dose and the second dose. For the Moderna vaccines, the interval is 28 days. The first shot helps your body recognize the virus and prepare an immune response, while the second dose strengthens the immune response. It's not unusual for vaccine to require 2 or even 3 or 4 doses. For example, vaccines given for children such as hepatitis B and diphtheria required several doses.
There are multiple COVID-19 vaccine existing so that other people can choose what vaccines they want. And every vaccine, there is different effect on the body. Health workers interview the person if they have illnesses, pregnant, and etc. in order for them to know what vaccines are applicable to them. Vaccines for COVID-19 are critical tools for helping bring the pandemic under control combined with effective testing and exacting prevention measures. How do the vaccines work? Vaccines teach our immune system to build a specific antibodies against diseases causing viruses and bacteria of their toxics. Your body becomes armed to fight these without ever having to become sick first. So in the future, when the body encounters the viruses or bacteria against which vaccines are made, it already knows how to defeat them. Several types of COVID-19 vaccines have been developed or are in development, including inactive or weakened viruses vaccines, which use a form of the virus that has been inactive or weakened so it does not cause diseases but still generate an immune response. Next, protein-based vaccines, which use harmless fragments of proteins or protein shells that make the COVID-19 viruses to safely generate an immune response. Next, viral vector vaccines, which use a harmless virus that carries the coronavirus proteins to safely generate an immune response. Last, nucleic acid vaccines use genetically engineered RNA to generate a protein that itself safely props an immune system.
Why there are so many vaccines in development? Typically, many vaccine candidates will be evaluated before any are found to be both safe and effective. For examples, all of the vaccines that are studied in the lab and laboratory animals, roughly 7 out of 10 will be considered good enough to move into clinical trials in humans. Of the vaccines that do not make it clinical trials, just 1 in 5 is successful. Having lots of different vaccines in development increases the chances that there will be one or more successful vaccines will be shown to be safe and efficacious for the intended prioritized populations.
Knowledge is one key to successfully managing your health. With vaccines being distributed across the nation throughout early 2021, it is important to understand the basics of vaccination. Not all vaccines work the same way. Traditional vaccinations like the flu shot typically introduce a small amount of a virus to the body. This causes you to build immunity against a specific kind of sickness. But thanks to scientific advancement, a different type of vaccination is on the horizon, known as messenger RNA or mRNA. These new vaccines are changing how you can stay safe from illness, including COVID-19.
Traditional vaccines work by helping your body develop antibodies, allowing you to fight off infections. Messenger RNA is a type of RNA that is required for protein production. In cells, mRNA uses the information in genes to create a protein for making protein. Once cells finish making a protein, they quickly break down the mRNA. mRNA from vaccines does not enter the nucleus present a promising alternative to conventional vaccine approaches because of their high potency, capacity for rapid development, and potential for low-cost manufacture and safe administration.
So the first traditional vaccine was developed at around the year 1800s. This led to many more vaccines being developed, thereafter saving millions of lives. And when these vaccines enter our human bodies, they tell our immune system composed of B and T cells to destroy this foreign material, remember what it looks like, and be on a lookout for future infections. Now, the mRNA vaccine designed for SARS-CoV-2 have changed this technology a bit. They look at the entire genetic material and then found a single instruction or mRNA that encodes one protein and decided to use this as part of the vaccine. When these instructions enter our cells, our own cells then make that protein and elicit the same immune response. They will destroy this foreign protein, remember what it looks like, and then be on a lookout for future infections. Now, scientists can use the traditional method of developing vaccines, but the problem is that the traditional vaccine is using a attenuated virus, or they are using the entire pathogens that have been killed or weakened so they cannot cause any more diseases. And vaccines that uses viral antigens are often less effective than vaccines that uses a whole virus. The antigen may not provoke a strong immune response, and if that particular antigen mutates, the vaccine becomes ineffective. So mRNA vaccines are much safer, more effective, but they are harder to make than the traditional ones. But the advantage of greater safety and greater effectiveness makes them unique.